Welcome, everyone. Welcome back to Anatomy and Physiology. In a previous video, we were talking about the cell. We talked about some of the organelles, and we really just focused on the structure of it. We didn't really get into the function. So in this short video, we're going to be talking about the different functions of all of these organelles. So let's take a look within that PowerPoint. So here is the plasma membrane or the cell membrane. And we said that it is that outer boundary of the cell. It's really designed to protect the contents within the cell. It keeps what's outside out and keeps what's inside in. And it is a phospholipid bilayer. We see here it's a permeable or semi-permeable lipid bilayer made of proteins and lipids, but we call it a phospholipid bilayer. The outer part of the cell is hydrophilic. The inside part of the cell is hydrophilic. And the inner part between that membrane where the lipids are is hydrophobic. Doesn't like water too much. And that's what makes the plasma membrane selectively permeable. It's saying, hey, you guys can come in, or no, we don't want you in, right? That's what selectively permeable means. It tries to decide what comes in and what should stay out. And one of the big things that does that are these proteins that are found within the cell wall. They make these passageways called channels. And the channel is really nothing other than a window or a doorway that allows things in and out of the cell. So certain channels will allow sodium in, or allow potassium out, or allow calcium in, or calcium out, or sugar in and sugar out. Each channel is specific for certain elements to go in and out. Okay. We went over the term extracellular fluid. We said that is the fluid that's found outside of the cell, whereas the cytosol was the internal fluid of the cell. The term cytoplasm, that's the contents of the cell. Everything that's outside of the nucleus, but before the cell wall. Right? It's everything between the nucleus and the cell wall. That's the cytoplasm. Now we have these structures that we can find on the cell wall, and they're called cilia and microvilli. Of the two, they're really like finger-like or hair-like extensions that come off the cell membrane. And the cilia are larger, and they're motile extensions, meaning they move and they can whip around like this. And they help to move the cell or to help move certain material throughout the cell. We'll find cilia primarily throughout the airways, like the respiratory tract, especially in the trachea and the windpipe. We have the cilia, and the cilia beat rhythmically. So if I were to draw here the trachea or the windpipe in your throat, we're going to have these columnar cells. They look like columns, so they're called columnar cells. And we're going to have them on both sides, these type of epithelial cells called columnar cells. And we have these cilia, these hair-like cilia that come off of it that I'm drawing here. Now, these cilia beat rhythmically. They beat superiorly up in this direction. Remember, air is coming down this way. You're breathing in and inhaling air down. So if there's dust particles or debris, the mucus that's actually sitting on the cilia trap the debris. And then they beat superiorly. They're beating upwards to push any debris outward. Okay. Microvilli are smaller than cilia. They're much, much smaller, right? If you look here, the cilia are 7 to 10 micrometers. The microvilli are 1 to 2. So they're very, very small. And when I think of microvilli, think of a sponge. A sponge. A sponge absorbs things into it. That's the function of microvilli. They're little sponges that bring in nutrients for absorption. So we're going to see microvilli all throughout your gastrointestinal tract, all throughout the intestines to absorb nutrients. Now, everything that we covered here, all of these structures contained within are referred to as organelles. They're specialized structures within the cell. They have a certain type of structure, a certain type of shape. They're all uniquely different, and they all have and perform specific functions in cell growth, in maintenance, and even in cell reproduction and division. Now, remember, we talked about the centrosome and the centrioles. Remember what we said the centrosome was? It was that large, dense sphere that contains the centrioles. The centrioles are these paired organelles that are really made up of a protein that are called microtubules, and they're pretty large proteins. And then we have the cytoskeleton, and the cytoskeleton is also made of proteins. Some are very small, they're very thin, some are intermediate size, and some are larger proteins like microtubules that make up the centrioles. So the cytoskeleton, just like the skeleton contained within the human body, provides structural support. So let's look at some of these functions. We went over the ribosome, and the ribosomes are a complex of RNA. What does RNA stand for? It stands for ribose nucleic acid. Ribose nucleic acid. So ribosomes are these complexes of RNA, ribose nucleic acid, and protein that are found in all cells. Now, the function of ribosomes is the synthesis of protein. That's really important. What does the synthesis mean? It means you're making or manufacturing protein. That's all it is. Ribosomes are responsible for making protein. Now, where do the instructions come from? Where do the instructions in terms of what proteins to make come from? Well, it comes from your DNA. Your DNA is the template of the instructions and the codes of what type of proteins need to be produced, but it doesn't actually make them. That's what the ribosomes do. DNA is different than RNA, right? DNA is deoxyribose nucleic acid. So we have ribose nucleic acid and deoxyribose nucleic acid. Now, remember in the picture before, in the earlier video, we were looking at ribosomes as those small white speckled dots, and we said that they can be free ribosomes or they can be fixed ribosomes. Free means they're floating free within the cytoplasm. Okay, they're floating free in the cytoplasm, and they can manufacture protein for the cell. Remember, ribosomes synthesize proteins. They make proteins. If they're free ribosomes, which means they're in the cytoplasm of the cell, they can make proteins that are used for the cell. Now, sometimes they're fixed ribosomes. Well, what are they fixed to? Well, they're fixed to the endoplasmic reticulum. And if they're fixed to the endoplasmic reticulum, they can make proteins as well, but their job is to manufacture the proteins that are needed for secretion outside of the cell. Right? Remember, I showed you that vessel, and we said it can move towards the cell membrane, open up and become part of the cell membrane, or it can open up and release its contents to outside of the cell, which is called exocytosis. So the fixed ribosomes can make proteins for secretion outside of the cell, that's exocytosis, especially if it's pushing out a neurotransmitter or an enzyme or a hormone, or it can be incorporated into the plasma membrane if it's just recycling the proteins. Okay, now on this slide, remember, I don't want you to confuse these two terms. They look alike, but they're not the same. Nucleus and nucleolus, right? Think of the nucleus. Think of the nucleus as a fruit, like a peach, and the nucleolus would be the pit within the center of it. Okay, so the nucleus, which is the larger, that's the control center of the cell, and that's where all the genes are located. And that contains your DNA, your deoxyribonucleic acid. The nucleolus, which is the pit in the center of the nucleus, that's the site of the ribosomal RNA, the ribosomal RNA synthesis. Now remember, around the nucleus is the nuclear envelope that has the nuclear pores, those holes. Well, it's the ribosomal RNA that's going to find its way out through the nuclear envelope, through the nuclear pores. Okay. Now the ER, the endoplasmic reticulum. The endoplasmic reticulum comes in two flavors, right? It comes in smooth and it comes in rough. The difference between smooth and rough is, does it have ribosomes fixated to it or not? Okay, if we go back and we look at that cell, the organelles, right here, we see that this part of the endoplasmic reticulum looks smooth, but this portion here looks rough because it has the fixated white speckles called ribosomes attached to it. So there's a difference between the rough endoplasmic reticulum, which we see here and here, or the smooth endoplasmic reticulum that's located here. If they look different structurally, they're going to function different. So what is the rough ER? It says it's continuous with the nuclear envelope, because it is. We could see it here. Here's the nuclear envelope, and right here, we see it's continuous with it. There's the rough ER, right? It kind of folds within itself like this. We have these folds here. So it is continuous with that nuclear envelope. Now, the rough ER has the ribosomes, the fixed ribosomes, and it's involved in the production of proteins, glycoproteins, and phospholipids. Where did we see phospholipids before? We saw it linked to the cell wall because it's a phospholipid bilayer. So that means your rough endoplasmic reticulum can make proteins, it can make phospholipids, and it could use that to regenerate the cell wall or any damage made to it, as well as glycoproteins. So these are all found within the cell wall. The smooth ER has no ribosomes. It's continuous with the rough ER, but this one does not synthesize proteins. Well, what's it going to do? Well, this one's involved in carbohydrate metabolism and the synthesis and modification of lipids, which are fats. So it's different, okay? The smooth ER is involved in carbohydrate metabolism and the production and the modification of lipids. What is the Golgi apparatus doing? The Golgi apparatus is going to modify the proteins. Well, it's going to modify the proteins that the rough endoplasmic reticulum produced, right? So it's going to like get the protein, repackage it, and then the Golgi apparatus is going to secrete that. As we saw over here, where this Golgi apparatus here released this vesicle that has a protein in it that is used to either recycle the cell membrane or to ship it out of the cell. Okay? The lysosomes. The lysosomes contain digestive enzymes. And if this is a lysosome right here, it's going to contain lots of hydrogen ions. So I'm going to put H+. So H+, these are hydrogens. Now, the more hydrogen that is found anywhere contained, it means that the pH, if this is the pH scale that goes from 0 to 14, and right in the middle is 7, anything from 0 to like 6.9999999 is an acid, and anything above 7.0 all the way up to 14 is an alkaline or a base. Okay, so our body's pH in terms of blood pH is maybe somewhere here, which is 7.4. But your stomach pH could be somewhere here, which is like 2.3, which is acidic because of all the hydrochloric acid we have in the stomach. But if we look at hydrochloric acid, we see HCl. H is for hydrogen, hydrochloric acid. The more hydrogens, the more acidic. So lysosomes are filled with lots of hydrogen and digestive enzymes. And when it finds something that may have invaded the cell, the lysosome opens up, releases those digestive enzymes and all the hydrogen, lowers the pH of whatever it is, and destroys it. It's really like cleanup. It's designed to rid the body of a lot of debris that's found within the cell. So that's the job of the lysosomes. Peroxisomes are a little bit different. Remember, the lysosomes were the yellow structure. They were smaller. I mean, they were bigger. If we look here, let me go back. So here's the lysosomes, the yellow one. That was a little bit larger. And the peroxisomes of the gray, they're a little bit smaller. Because they look different, they function different. The peroxisomes are enzyme-filled vesicles, much like the lysosomes were, but the peroxisomes are going to detoxify harmful substances. It's going to neutralize free radicals, and they're found highly concentrated in the liver. So they're really good at detoxifying alcohol, drugs, things that are harmful to the body. So it puts, since the liver is highly concentrated with peroxisomes, that's why when someone consumes a lot of drugs, medications, alcohol, someone can get liver failure because it stresses out these peroxisomes. And there's no more peroxisomes with these enzymes available to help neutralize, detoxify, and neutralize these free radicals, which harm your DNA. And it's also involved in something called beta oxidation. When you see the word beta oxidation, that is breaking down of fats and fatty acids. And it does so, beta is which carbon, like the first is the alpha, the second carbon is the beta. So you look at these long carbon chains, and in order to break them down, it chops it at the second carbon. They call that beta oxidation. Okay. And the last organelle is the mitochondria. And this is the site of energy production. And energy production, the energy source within the body, or the energy currency of the body, is called ATP. And ATP stands for adenosine triphosphate. So these mitochondria produce ATP, adenosine triphosphate. And that's because with the presence of oxygen, the oxygen can oxidize sugar or glucose and start to produce ATP. Within the mitochondria, in your lecture, you will learn about the Krebs cycle, and you will learn about the electron transport chain. The Krebs cycle and the electron transport chain are involved in ATP production, and both of them take place in the mitochondria. But they're dependent on oxygen, and they're dependent on even glucose and oxygen. Now, where does oxygen bind in the body? Well, it binds to red blood cells, right? You have blood cells that have hemoglobin. And heme is iron. And iron just doesn't float freely within the human body. It has to bind to a protein, right? One of the functions of proteins, you will learn, is that they're referred to as protein carriers. They move something safely within the human body, in this case, iron. So it's called hemoglobin. Globin is the protein that binds to heme, which is the iron containing portion. And iron is the metal that binds to oxygen. And that's why if someone's iron deficient, like iron deficiency anemia, they don't have energy. They fatigue because there's no iron to bind to oxygen. You don't have oxygen, the mitochondria can't produce ATP. And you're tired. Think about exercising. You're at the gym or you're running and you're doing some form of exercise and you're breathing heavy. You're out of breath. You got to stop because you need oxygen to replenish ATP. So once you stop doing what you're doing and all of a sudden you're not in an oxygen debt anymore, but oxygen levels rise, the body can regenerate and produce more ATP so you're not fatigued. So this is where the Krebs cycle, known as the citric acid cycle, and the electron transport chain take place. And again, the Krebs cycle or citric acid cycle is dependent on a lot of your B vitamins, especially B1, B2, and B3. And the electron transport chain is dependent on iron and CoQ10, coenzyme Q10, to help produce ATP. Now, here's a clinical gem for you, for those that are interested, or maybe you have family members that are on cholesterol medication, or grandparents or parents, or maybe some yourselves are taking cholesterol medication known as statins. What the statins tend to do is deplete the body of CoQ10. And if they deplete the body of CoQ10, they're going to deplete the electron transport chain ability to produce ATP. Now, the question is, where do we find most of the mitochondria? We find most of the mitochondria where there's a lot of energy production taking place, meaning what parts of the body are always working that need energy? How about your muscles? How about your heart that's always beating? How about your brain that's always functioning, your nervous system? That's where we find the most mitochondria. So if the cholesterol medication or the statins like simvastatin, Crestor, Lipitor, these are statins, if they deplenish coenzyme Q10 and it depletes the body's ability or diminishes the body's ability to make energy, then the mitochondria suffer And the parts that have the most mitochondria often have the symptoms. So people get muscle aches or muscle pains. They call it fibromyalgia. The heart starts to malfunction. You get these cardiomyopathies, or the nervous system starts to dysfunction in the brain, and you get cognitive decline or dementia-like symptoms. There's always an explanation for things. And when you understand and look deeper into the physiology of how these medications and drugs work, it can help offset some of the adverse reactions. Okay, so that's some great information there. Now, the other great information about the mitochondria is that they resemble and look like bacteria. They resemble and look like bacteria. And when an individual uses or overuses antibiotics, antibiotics go after anything that's bacterial or that even looks like bacteria. So oftentimes they destroy many mitochondria, which diminishes your energy production. So when people take antibiotics, they can actually hurt their mitochondria, and some antibiotics damage the mitochondria so much you often end up with muscle tears and tendon tears. There are certain forms of antibiotics that classically rupture the Achilles tendon. So you have to look at those forms, look at adverse reactions and what they replenish, and see if they can cause myopathies or even tendinopathies, something to be careful of. Okay.